Okay, so histone modification is really useful if you don't know the transcription factor. That's important a priori, right? You just profile, and then after that you use the motif analysis. Another technology was developed a little later after the histone modification, and it's actually also very, very useful. This is called the chromatin accessibility profile. Initially, people use something called the DNase. So basically, DNase is an enzyme that can just cut the genome. Theoretically, if you have enough DNA, it will chew up the whole piece of DNA into only mononucleotide, really. But if you don't give the chromatin enough of this chewing, you only have a little bit of DNase and a lot of chromatin, DNase will go wherever it's easy to go. But where is it easy? It basically is open chromatin, nucleosome-free regions when transcription factors are there, or the region has a lot of histone marks, the good histone marks that are open. And so DNase will go there and chew. And so if you don't have enough of the enzyme, it only just make occasional cuts. By chance, a lot of the fragments are different. You run this DNase digestion only for a short time, and you run it through a gel, you will see kind of a smear. A lot of fragments are really long and some are a bit shorter, but then in this experiment, you can select the really short fragments that are only maybe 100 or a few hundred base pair long. And this means that this region is somehow very open, that the DNase happen to cut. It make two cuts because one is before and one is after you are selecting the fragment in the middle. And there are two cuts that are fairly close to each other within 100 or 200 base pair apart. And so you can just isolate those short sequence and then sequence them, map them to the genome. And this is again a sequencing experiment from a DNase hypersensitivity experiment. And you can see here, most of the region of the genome is just some random. There are occasional cases that two cuts are close by, but the signal is quite weak. Whereas when in those regions, when transcription factors are binding there, you see very, very strong peaks in there. And in those peaks, you also notice a little bit, the plus strand reads are blue, the negative strand reads are yellow, and usually the transcription factor really lands in the middle. Right? So you get very, very good enrichment. And all of those peaks, they tell you transcription factor is binding in those locations. It can be either on the promoter or on the enhancers. And this is a study published by John Stam's group from the ENCODE consortium. And so they did a DNase hypersensitivity assay on this cell. You can see this is the, the roughly region of the chromatin. You can see these are where the peaks are. And it happens that in this cell, scientists have done transcription factor ChIP-seq in many different transcription factors. It's all in the same K562 cell, but the ChIP-seq, this is on the ATF3 transcription factor, this is on the CEBP beta transcription factor, this is on the EGR transcription factor. And when they collapse, basically they added all the reads together from all the transcription factors. See, they created this combined transcription factor density. You can see it overlay really, really well with DNase. And that's really great. It means that if you can do DNase-1 assay, well, potentially you don't need to do, all of those different transcription factors, right? You just do this one, at least these are all the shadows on screen. The transcription factor only go to those locations, and any transcription factors should bind to this. And that's why if you go back to the previous transcription factor ChIP-seq quality control study lecture, we mentioned that a way to know whether your ChIP-seq data is good quality is you get those peaks and you ask how many of those peaks we have seen in a DNase hypersensitivity study. Most of the time you do a transcription factor ChIP-seq, you get a few thousand peaks, 80 to 90%, or even 98% of the peaks overlap with some DNase before. because the DNA is collected all the, every cell you do a DNase, you get maybe hundreds of thousands of peaks. Another cell you do DNase, you get hundreds of thousands of peaks. You look at all of this, a transcription factor binding should really overlap with all the DNase hypersensitivity peaks. And so having high overlap with the DNase is another indication for quality of your ChIP-seq. Okay, so if you see this region having high DNase hypersensitivity, it means that potentially the transcription factor that can bind there is binding pretty stably and is binding in a lot of cells. Whereas if this region is kind of only a little bit of a peak, that means the transcription factor binding is kind of weaker. It's not in all the cells, in maybe a small percentage of cells. It really can tell you also binding stability, but this stability can be due to the binding of one transcription factor or many transcription factor binding all in that location together. Okay, so that's kind of the DNase hypersensitivity peaks. And by the way, for this, you can also look for transcription factor motifs, right? So if you do a DNase, you see what are the differential peaks between a disease state versus a normal state. You get those peaks, you look for motifs, you can see what transcription factor binds there. It's a very important way. So the original study, the authors also want to look at something called the transcription factor footprint. By the way, these peaks, the highest resolution peaks is either the transcription factor itself, because it's only binding by, like a few nucleotide long, or DNase. Histone modification peaks, even the really sharp ones like K27 acetylation, is not as sharp as the DNase peaks. DNase peaks are usually only, 150 to 200 base pair long. It's really sharp, which is really good. But then the developer of this technology, they also want to use something called the DNase footprint to look at transcription factor binding in even higher resolution. And so they look at some locations, for example, if they see a DNase peak in here, but then they really look at not just the peak, they look at where exactly this read lands in this, like a 200 base pair location. They zoom in to see where that read lands. And they saw a case where the read is to the left, and there are reads that's really marking the right, but then in the middle, there is a gap where theoretically the transcription factor is binding in there, it's occupying this location, so it's protecting the chromatin from the DNase cutting. And so you will have a cut here, you might have a cut there, you might be isolating the fragments, you're getting the read from the left and right, but not right in the middle. And so they are using this approach, a footprint, to really look at where the transcription factor really binds, which is really exciting, right? You can use it, you can know the transcription factor binding even to the base pair resolution. The danger of this is the specific way they look at this is they say, if there is DNase signal, if we look at the left end read and the right side, or the read that lands in those locations, if there is a gap in the center, that's an indication there might be some transcription factors binding there. And they specifically have a footprint occupancy score where you can see they look at the C plus 1, because they don't want to have 0, divided by the reads on the left, and the C plus 1 divided by the reads on the right. By the way, for L, C, and R, they test between 6 nucleotide to 25 nucleotide. They try everything. And then they ask which one do they get the lowest kind of a score, which means that the left and the right is very high, but the center, suddenly you see a trough. That's an indication the transcription factor is binding in the middle, and that creates this footprint. But so one thing we need to be actually very careful about this is DNase actually have cutting bias. So we look at these properties, even with naked DNA, which doesn't have chromatin. There's no nucleosome, there's no chromatin, it's just double-stranded DNA. You use the amylase to make the cut. It will still have this distribution, which means that some cuts are more likely to happen in other cuts. And so because with DNase, you know every time you see a read, it means there is a cut to right next to it. So we can look at every time the read map there, and we look at the sequence there, and also the 3 nucleotides before this, we can build a cutting model. You can see, for example, some regions can be cut quite frequently, whereas other regions may not be cut at all. And so the intrinsic DNase cutting bias can have as big a difference as 300-fold, which means that sometimes you might have a left and right, which is very high, and the middle that's really low, but that's caused by a cutting bias. You know, the DNase enzyme just like to make cuts in here and likes to cut in there, and then in the middle, it couldn't cut very well, and then you would have a gap. And so, so far what we have found is that actually using the footprint to predict the transcription factor is actually not a very good approach. It doesn't really give you better prediction. Instead, if you only just use motif in the DNase peaks to make prediction, you actually are doing a better job. And so this is a study that was, you know, originally published. They say, oh, so they say, see, there's a peak in the left, there's a peak in the middle, and there's a gap in the middle. Sorry, a peak in the left and right and a gap in the middle. They say, there's a transcription factor binding there. And they can see it not only in human, they also see the same sequence in mouse having this cutting. There must be a transcription factor binding in this location. It turns out this is actually a bias. If you even look at the genetic DNA, it's because of the DNA. If you just look at the genetic DNA cutting, naked DNA, it also creates this, you know, left cut and right cut, and the middle is unfavorable in terms of cutting. And the motif they identify there is not a transcription factor motif at all. So we need to be more careful about this. Okay. Questions about DNase footprinting. So by the way, the world expert for DNase hypersensitivity experiment is John Stam's group in University of Washington. He has generated DNase profile for thousands of different cells in different, you know, developmental stage, disease, different cell lines. And his data quality is really, really great. And this data is very, very useful in general, because anytime you are interested in some cell, you can look at his data, at least you know where the shadows are and where you should focus your attention on. Okay, so that's a DNase.